So yesterday's lecture was really nice.
Yeah, yeah. So he has done a lot of work.
We got some similar gene, maybe
Okay,
completely different in sequence, but
okay,
again those are 54 amino acid longer.
okay, okay.
Only.
So we are already in YouTube live, and so we should start
Sure.
this session. So good afternoon, everybody. So we start the today's second session. So it will be 3 to 5 p.m. Every speaker will be having 1 hour, where 50 plus 10 minute so presentation will be there. So we start with the first presentation, Dr. Amolkumar Solanke. Second speaker will be Dr. Kutubudin Molla. So and for this session, we have the panel member from our department, Professor Vidya Devi Negi. So she will be handling this session. So Professor Vidya Devi Negi, please.
Thank you very much, Dr. Vinod. So, on behalf of the department and whole organized team, I welcome you all for this session. And in today's session, we have two speakers, as already Dr. Vinod has mentioned. So, the first speaker we have Dr. Amolkumar Solanke from Indian Council of Agricultural Research, National Institute of Plant Biotechnology, Delhi. So, Dr. Solanke, did his PhD in plant molecular biology from Delhi University in 2009, and he has been working as a scientist at ICR, National Institute of Plant Biotechnology, New Delhi. And his research used genomics and bioinformatics approaches for biotic and abiotic stress management in the crop plants. So far, he has completed various projects to number around 11 to 12, and the funding agency from SERV, DST, ICR, etc. as a PI and co-PI. So he has published a numerous number of papers like 55 and 12 book chapter and 12 manual and edited chapter. And he has been working as a faculty of Indian Agricultural Research Institute, New Delhi, and also he has worked as a faculty at Jamia Hamdard University, New Delhi. He has guided around 6 MSc students, one PhD student has been passed out, and currently 4 PhD students are under his guidance. He also has served as a reviewer for various international reputed journals like PBJ, Scientific Report, BMC Genomics, Frontier in Plant Science, and many more. These are highly reputed journals, so it's a very good contribution he has done. And in 2016, uh, he has filed a patent also for his research work, which is going in his laboratory. And he has various awards also in his credit, like Jawaharlal Nehru Award, which he received in 2012. He is also recipient of Eminent Plant Biotechnologist Award 2015, Excellence in Research Award 2017, Fellow of Society for Applied Biotechnology 2014, and C.V. Jacob Award 2013, and Young Scientist Award and ARS Scientist. So these are highly prestigious awards, and his expertise and scientific area include use of functional genomics approaches for blast resistance in the rice. So we look forward to have a very wonderful and a very interactive talk from Dr. Solanke. So his title for his talk is Understanding Rice Magna Porthe Interaction During Panicle Blast. So Dr. Solanke, please, so you can share your slides.
Sure. Okay. So good afternoon all. Uh, Respected organizers, thank you for giving me this opportunity. I am Amolkumar Solanke from National Institute for Plant Biotechnology, New Delhi. And here I am going to talk about actually what we are working in our lab, that is regarding panicle blast. So title of my talk is Understanding Rice Magnoporthy Interaction During Panicle Blast. Okay. So there will be questions in between or
At the end,
No, the
we,
question,
end only?
yeah,
Okay.
question will be in the end. So from both the panel and from the YouTube. So in the end, we'll have the question session, sir.
Okay. So basically, if we talk about rice, it is the we will say staple food of India. And in India, almost 44 million hectare area is under rice, and its total production is 167 million ton. And we know that this rice blast is one of the most serious disease of rice, and it loss sometimes directly and indirectly. And its annual, if we see, total production loss is from 10 to 30%. And here, if we see, almost all part of rice get affected by this blast. But out of this, especially when this panicle get affected, at that time, its losses are up to 100%. It can lead up to 100%. And it is, we will say, it is almost pandemic that it covered all world. Okay, but if we see, there are some chemicals which can control this blast, but most effective way to control this rice blast is host gene manipulation through introduction of blast resistant genes in cultivated species. And till date, almost 100 of rice blast resistant genes has been mapped, and out of which, if we see, 21 genes has been clone. Out of this, there is PI54 gene, which was cloned and characterized in our own lab by Dr. T.R. Sharma, who is now DDG Crop Science. Uh, but, and if we see here, out of all these 21 gene, except one gene, all are NBS LLR gene. So there is importance of NBS LLR gene in blast resistance. And there are some other genes also, which we will say defense regulator genes. And these defense regulator genes are not our genes, but still they are providing resistance to blast. Okay. And if we see wild rice reservoirs, these are the good source of these resistant genes. And mostly this Rufifogon, Rhizomatis or Officinaris, this lines has been used to get these genes from for resistance. And most of the genes like PI9, PI40 or PI54 RH or PI54 OF, these has been characterized from this wild rice. So first we decided to identify these genes from available data. So these are the total 11 wild species of rice. Its data were available and all are from AA genome. So which are closest to our Oryza sativa. So we predicted this NBS LLR genes from this one, and surprisingly we observed that most of these genes are present on chromosome 11 followed by chromosome 12 in almost all 11 species. So we identified, then we also identified different domains of this NBS LLR genes along with this NBS LLR, and we also identified its paralogs present within or orthologs among different species and species specific genes, so that there are some genes which are present only this particular species. This is the map, genetic map, on which this species specific genes has been put on this map. And again we observed that on chromosome 11 most of these genes has been identified. And then we identified these genes are really species specific or not. So there are some genes like from Bharti or Nivara, these genes are really species specific. These are not present in other species. We published this in Plant Molecular Biology report. And after that, we actually start identifying genes from same cross, because this TTEP, this is really a big source of these resistant genes for blast. So in 2005, as I told earlier, Dr. Sharma, who is DDG, I identified this PI54. Earlier he gave name PIKH, and now it is PI54, and this is broad spectrum gene. So this particular gene is activate, it is inducible gene, it activate, and further all complex defense mechanism which we have characterized in one of our transcriptome data. And further this robust DNA markers has been developed by him, and further this has been used, and now almost 26 varieties has been in the process of release, and most of varieties are released. So these varieties in which this PI54 is present. So now after this, where they have developed this TTEP into HP22 cross and 128 real population recombinant inbreed lines. Now it is in almost generation F15. So these lines are available with me. So what I did, I sequenced these both parents, TTEP and HP22, and further genotype this all real population with 50K SNP chip. So our Professor N.K. Singh sir is there in our institute. He developed this 50K chip using single, we will say single copy genes identified, and using those single copy genes, he developed these 50K chips where 50,000 SNPs are present on this chip across all 12 chromosomes. So we genotype this 128 lines using this one, and further I did transcriptome at panicle level. So panicle blast level at 3 different time points. Okay, so we developed and this transcriptome in both parents, that is in TTEP and HP22 at 0 hour, then 48 hours, 72 hours, and 96 hours, so that we can get the how this orchestra of genes is present during panicle blast. And this is one of the example where how we can identify using 50K chip. So on the basis of parent, and one of the best line, this is real 4 line, which is almost similar to HP2216, but having resistance from TTEP. So this line we have identified, and here red is shown by TTEP and green is shown by HP22. So like this, we can identify which genome is present using this 50K chip. So this particular real 4 population, which is best, is crossed with again HP22, and where we identify there is duplicate gene interaction. So interaction is 15:1. So we check it in F2 derived F3, where 3 different progeny, 1 progeny 2 and progeny 3 we have characterized, and this is in Malan. And if we see here at this is blast nursery, where if there is blast, it does not survive. And in panicle blast, you can get this black color in the neck. So like this, all these lines we have checked with SSR markers, almost 109 SSR markers, out of which 49 we found polymorphic, but none of this go with this blast phenotype. We also check these ISSR markers, although these are not good markers for checking, but still we got this one marker which is associated with this one. And here if we see this particular band, this band is present in resistant line and absent in susceptible line. Okay, there are few we can say crossovers are there, but still there is pattern is similar. So we pick this one and we sequence this particular marker, and then we observe that it is on chromosome 12. So now, since we have this 128 lines, we try to map this particular region in which number of these NBS LLR genes are there. So now we need to do fine mapping of this one. But we were facing problem while working on panicle blast, because in Delhi condition, we don't get panicle blast actually, because panicle blast need high humidity and low temperature. So this particular season we don't get in Delhi, and we don't get. So that's why we decide to develop proper method so that we can get panicle blast in Delhi condition. So we are growing this magnoporthe on PAD after up to 15 days, and after that we are transferring it on Mathur media, and from Mathur media, when these pores appear, after that we are making suspension so that it should be 10 to the power 5, and after that we are injecting it in the neck of this panicle. So like this, and after that we are putting cotton plug with water, and then we are attaching this aluminum foil. so that this water should be there at least for 24 to 30 hours. And then we are keeping it as such, and after almost 21 days we are checking whether there is blast or not. So we are getting clear cut variation in resistant and susceptible line. So in susceptible line, if it is really severe, we don't get any panicle, and in resistant there will be only mark of injection. So but using this we can screen all lines. Okay, so using this we are check in TTEP HP22, these are resistant line, and HR12 and HP2216, these are susceptible lines. So, and whether there is really panicle blast we are getting or not, we check it using PR genes. So here we check in both TTEP and HP22 at 48 hours, 72 hours, and 96 hours, and we check that this PR2 and PR3 both are upregulated in resistant line, but not much in susceptible line. Still there is some upregulation. Okay, so we can even differentiate on the basis of this whether these are resistant or susceptible lines. But here PR7, PR10 and PR14, here expression of these genes are almost similar in resistant and susceptible line. So we confirm that yes, using this method we can get panicle blast properly. And after that we screen almost all wild rice accessions which we have available here, and out of which here you can see different type of pattern we can get, from complete resistance to susceptibility, different level of susceptibility, that is in this 403 complete panicle get yellowish using this one. So like this we can get pattern, and here we did not check it with only one strain. So we check it in this all lines with 3 different strains, that is Sikkim, Dehradun and N22. Okay, and we identified where we got susceptibility with all 3 strains. Now we called it as susceptible, and where we called got resistance with all these strains, we called it as a resistant line. So like this, now our future plan is to check all these lines with more virulent strains of Magnoporthe, so that we can get best line to cross with and to get important genes. Further, we also did the analysis of this one with different genes. So this resistant lines and susceptible lines we check. Then we give for mock 24 hours and 72 hours with 3 replicates and isolated RNA from this one. And then check this some important genes which we have identified through panicle transcriptome. I am in coming slides, I am going to discuss about how we have characterized this panicle transcriptome. Okay, so these genes where this 13 and 396, these are susceptible. So randomly we pick to identify, can we identify some marker genes through which we can say yes, it is really resistant or susceptible. So out of all these, we got only this NBS-LLR. This is one marker where this particular gene at 24 hours, it is simply one-fold upregulation, or we can say there is no upregulation of this particular gene in susceptible lines, but there is upregulation from 2 to 5-fold in resistant lines. So if we are using this as a marker, we can identify whether this particular gene is, this particular line is resistant or not for almost all strains. So we are trying to identify these common markers using this analysis. And till date, only single gene has been identified by Japanese group, that is PB1, panicle blast 1. This is the single gene. And again, this gene is from Modon. This is indica variety, and from which it has been transferred to Japanese varieties. And these PB1 plus varieties, these particular varieties, there is no breakdown up to 50,000 hectare paddy, and that too for 30 years in Japan. So this is really, I will say, broad spectrum and durable resistance this particular gene is providing. and this gene has been generated in this particular line, modon line, because of gene duplication and formation of new promoter. So that's why we decide to characterize this particular gene through allele mining, whether we can get this particular gene in other wild type. So we take this 2 kb of promoter and 4 kb of this gene, and almost 6 pairs we have developed, overlapping pairs, so that we can get complete gene. So we did analysis in almost 55 uh, lines from these 5 different rice accessions: Oryza sativa, Indica, Nivara, Bharti, Rupifogan, and Glaberrima. And we found that almost these lines which are resistant, which are falling, resistant and susceptible, are falling in different groups. So we identified this one, and again we check expression of this, as this PB1 have more expression in panicle. So whatever lines we have checked, either TETEP, wild rice 2 or wild rice 398 or 430, their expression of this particular gene is more in panicle as compared to this. There are some exception like CO39 where there is slightly more, but it is not reaching to at least double in panicle in this resistant line. So like this, we have identified these lines, and from this we can take these lines where there is more expression of this particular gene for further crossing. But we also try to identify other sources. So for that, what we did, there is a mutant population of Nagina 22. So EMS mutation is given. So we have taken M2 population of almost 1.5 kg, which contain more than 1 lakh seeds. So we sown that particular seed in this Hazaribagh. So there is a regional center of Cuttack, Rice Institute Cuttack in Hazaribagh. So where these plants has been grown in natural nursery, or where this natural occurrence of blast is there. And from there we have selected 60 lines. So out of 1 lakh plants, we have selected 60 lines with score of 1, 2 or 3. And then again next year also in 2017, next year also we have shown those 60 lines there, and we screen these on the basis of this one. Here score was 0 to 9, and out of this we have selected only 1, 2 and 3. And in next year we got 0 to 3 source, and these are really resistant line. So 2 years we have selected there, and after that we bring those lines here and we sown in Delhi condition, and after that we check all those 60 lines for panicle blast as well as lip blast in Delhi condition. So these are my students who are continuously working almost for 1.5 months every year to check these lines for this resistance. So we are checking almost 5 plants from each line and 3 panicle from each plant. So we check these lines. So like this, we are injecting. This is a needle, and this is cotton plug where we are injecting in the panicle, and after injecting in the panicle, we are putting this cotton plug. So we are checking this line. So we are getting proper blast here. So this is susceptible parental line Nagina 22, and these are mutant lines we got. So there are only marks we are getting in some lines, and otherwise in some lines even there is no mark at all. So we are checking and we identified few lines using this one, resistant and susceptible. Here we also check in under control condition. We have given panicle, we are spraying this magnoporthe, and after that we are checking individual lines so that we can get for both. So here also we identified some lines in this red color. These are resistant lines we have identified through this in our rice blast phenotyping facility. We also gave this material to northeast hilly regions of Ummiyan, and there also they have checked for lip blast. And after that, using all this, we identified this 13, 16, and 56. These lines are resistant for lip as well as panicle blast at all three centers, Hazaribagh, Delhi, and Meghalaya. And now we are trying to characterize this particular material. So this is one story. Now we have started analyzing transcriptome of these two lines, TTEP and HP22, at three different time points. Okay, so here we use same method of injection, and then we have given along with control, we have checked this. This is resistant and this is susceptible line, and this is the methodology where we are growing this magnoporte, we are injecting, and after that, after control, then after 48 hours, 72 hours, and 96 hours, we have isolated RNA. We check this quality of RNA. We after QC, we have sequenced this one. So after QC, we have sequenced this, and we got almost 9.5 to 10 GB data for each sample. Okay, and then we did differential expression. So using FastQ, we have identified these quality rates and then trim these rates. After that, we use alignment using top hat, and using cuff link, we have assembled. Further, we did cuff merge, and using cuff dip, we have identified differentially expressed genes. Okay, so and here we analyze all those genes which are only differentially expressed as compared to its control. Okay, so here when we are saying T48 means TTEP48, it means these are differentially expressed genes when we are subtracted with control. Okay, so we have identified all these genes with control, and then surprisingly, we observed that genes expressed in susceptible line and genes expressed in resistant lines, even upregulated and downregulated, are altogether different. So there are very few genes which are common. So if we see here, only 30 at 48 hours, only 37 genes are commonly upregulated in HP22 and TTEP. And here 69 genes are common which are downregulated in HP22 and TTEP. But if we see here, almost genes which are in resistant and susceptible, these are specific. And this trend is almost at 72 hours and 96 hours. We check these common genes which are expressed there, and we observe that in almost, when we compare all 6 conditions of TTEP and HP22, we found that 341 genes which are upregulated in all 3 conditions like TTEP 48, 72, and 96. Okay, and there are 384 genes which are common in susceptible 3 conditions. So we have identified even in upregulated separately and downregulated separately. Then we did annotation, geo annotation, and surprisingly we found that genes in all biological processes, molecular function and cellular components, these are more in TTEP as compared to HP22 in all conditions, okay, and in all 3 geo annotation. But at 48 hours, 72 hours and 96 hours, more genes are present in HP22. So it means early expression of resistant genes are more important for giving overall resistance as compared to these 2 conditions where there are late expressions in this HP22 as compared to TTEP. And then we identified these pathways. Okay, so in these pathways, we check these genes in HP22 and TTEP at same time point. And here we use only differentially expressed genes. So here if we see, the signaling, especially we observe that cell wall related genes are more as compared to this HP22 in TETEP. And here, if we see signaling related genes are also there. So these signaling related genes, cell wall related genes, and again secondary metabolite related genes and transcription factors, more number of transcription factors are upregulated in slides. And this particular trend is common in coming hours. So this is for 72 hours. Here also signaling related genes are more upregulated in TETEP. Here also cell wall related. So it means there's somewhere cell wall is functioning properly, or cell wall related resistance is, I think, major cell wall strengthening genes are expressing, and these are providing resistance in this panicle blast in this TETEP line. And here, these genes are also more, or transcription factors activation is also higher in TETEP. And this trend, where in 96 hours, although number of genes we have seen are less in this one, but these important genes, upregulated genes, are more for signaling here. Then even this jasmonic acid and other related genes are more here, and cell wall related genes. So these genes and secondary metabolite related genes are more expressed in TETEP. So this means these are more mechanisms or more pathways which are contributing for resistance in this TETEP as compared to HP22. Then we have selected some genes using this one, and we identified these genes. We check it for expression analysis. And this particular gene, which we observed that brassinosteroid insensitive associated receptor kinase, this particular gene we have selected for characterization using overexpression. So when we check using semi-quantitative, surprisingly we observed that in TETEP there is no expression of this gene, but it is slowly upregulated at different time points. But in normal condition, this particular gene is upregulated. These genes are upregulated, but because of infection, there is down regulation. It means in this HP22, there is something, or magnoporthe must have something which is suppressing these genes, which are really important for resistance in this line, cultivated line. But somehow, since it is not able to block in TETEP, so these genes are overexpressing and probably providing resistance under this planicle blast condition. We also check it through QRT PCR, and we observe clear-cut upregulation of these genes. Like this is ethylene responsive gene, which is upregulated many-fold as compared to this susceptible line. Okay, and this trend almost we observed similar to transcriptome, and where we have got some genes which are upregulated in susceptible line. So these are really upregulated here. This is cysteine-rich gene. This is upregulated only in susceptible line. So probably this type of genes are responsible for increasing susceptibility in this line. Okay, so from this, we have given this mechanism that whenever magnoporthe spores attach to this particular cell, what is happening in this cell wall and cell membrane, when it's entered to the aprosorium, there is signal through all this change in carbohydrate molecules. It is passing inside the cell, and there is complete ROS production. And because of ROS production, the signal is detected by kinases. After that, through transcription factors, these defense-related genes get activated. And these defense-related genes like chitin, PAL, or lacase, or glucase, or PR genes, which are activated. And further, this particular pass information and these antimicrobial compounds get activated. And this finally lead to the this programmed cell death, and which is ultimately gives hypersensitive response. So this is the mechanism happen in TTEP line. And we got many more genes which are still uncharacterized, and these genes are structurally different NBS LLR genes. Then there are some specific transcription factors which are upregulated only in resistant line. Then there we have identified some genes and which we characterize on the basis of few domains which are present in this, but otherwise these genes are novel genes like VWF. So this is Van Wilborde factor, which is uh, this particular gene. I like to talk about this gene. So this particular gene is upregulated when there is blood clotting during when there is a cut in human body. Immediately this particular gene get activated, and because of which there is blood clotting. So now we hypothesize and we are characterizing this particular gene that when there is magnoporte spore attached here, because of this particular gene get activated, and probably because of glycosylation, it is forming one net and it is not allowing magnoporte to enter inside. So at cell wall level only, it is providing resistance. Then there are eukaryotic aspartyl proteases gene. There are some domon genes. Then PMR, that is powdery mildew resistant 5 gene, or blufensin gene. Yesterday, Bhattacharya sir discussed about this DR1 protein. So we also got in rice similar type of these. These are blufensin genes, and again there are only 3 copies of this gene. So it is just 54 amino acid protein. So this particular protein is when it get activated in resistant line only. So out of this, so probably it may also need to be characterized properly, and we are doing this one. And as BAK genes which we are characterizing, this is kinase. So these are some novel genes which we have identified using this study. So as I told, this is structurally different NBS LLR genes, and why we are saying it as a structurally different. So when in integrative genome viewer, we identified this one. So these 3 lines are from TTEP, and remaining 3 lines are from HP2216. And when we check as compared to this MSU rice genome annotation release 7, there are 2 genes present separately, okay, in Nippon bear, and also we observe this type of gene in our susceptible line. But when we compare, there is complete gap between these 2. And again, here I need to tell you the data which I am showing here, it is actual expression data. We mapped these Illumina reads here from transcriptome. So here we are getting gap, and when we observe that this is complete gene, so it means instead of these 2 genes, we are getting this complete gene, and these genes are probably responsible for resistance in this TTEP line. So like this, we got 6 lines, 6 different genes. Okay, so these are codes. Sorry, don't go with these codes, because since we are characterizing these genes, I have not given actual numbers. So these are codes. So here, this is one gene which is completely novel gene which is not present here. Then this is another gene where along with NBS LLR, there are more different domains are attached there. Okay, here this is combination of 2 genes we observe here. Here also it is combination of 2 genes where along with NBS LLR, Verki is there. Here along with NBS LLR, there is kinase is there. Here DW40, DW40 domains are there, or P502CC domain is attached here. And here it is combination of 3 genes actually. So 3 genes are combined there, and along with NBS LLR, 2 Verki domains are attached here. And here again one novel gene is observed. So somewhere if we see this particular gene, there is all together, all of a sudden there is stop codon, but it is this stop codon is converted into normal, and this particular gene is extended further. So like this, these are completely structurally different genes, and we are characterizing these genes through transgenic approach, so that we can directly get evidence that yes, this is the gene which is providing actual resistance. Along with this, we also try to identify susceptibility related genes from this data. And if we say susceptibility related, these genes are responsible for successful pathogen infection. And these are essential for compatible plant pathogen interactions. So these three genes were already identified and characterized: PI21, OS MAP kinase 5 and SPL11. So along with this, we have identified from our transcriptome data 6 genes. From this one, these are the genes which are upregulated only in susceptible line as compared to resistant lines. And then we check its expression using this integrative genome viewer. And here, if we see in these susceptible lines only, these are upregulated as compared to resistant line where there is no expression or very less expression. And further, we check its expression through QRT-PCR, and we confirm that this red and this one, this is susceptible, insusceptible. The line, its expression is very high as compared to resistant line. Then we use CRISPR-Cas method to characterize this one, but instead of characterizing single gene or mutate single gene, we decide to characterize multiple genes in one construct. Because if we take multiple genes in one construct, each gene will carry almost 400 to 500 base pair. So if we are checking almost 6 to 7 guide RNA in one construct, it will be very bulky. So we decide to use natural RNA processing system. So here in between tRNA, there is gap or spacer. So we converted this guide RNA we transfer the spacer with guide RNA, and we make it in such a way that natural endo RNAs, RNase-P and RNase-Z, it will cut this guide tRNA separately, and remaining portion will remain as a guide RNA, and this guide RNA will further go and start its work. So at a time, if we see here tRNA plus guide RNA, only 214 base pair will be there for each guide RNA. So like this, we can clone number of guide RNA using this tRNA guide RNA system for multiplex editing. So we prepare this guide RNA, 2 guide RNA from each gene, and further using golden gate assay, using this PGTR, where there is already tRNA guide RNA scaffold is there. So using there, we have amplified this one, and further we clone using this golden gate assay. We clone all this one. So we got this type of after PCR amplification and golden gate assay, we clone this in PGEB32. So here in this we clone 5. In this case we clone again 5. Here 4 guide RNA and here 4 guide RNA for 2 genes. So like this we confirm here, and again this should be our vector should be like this. So we prepare this vector, and again we sequence this, and we confirm that yes, whatever guide RNA we are talking for these different genes, PI21, then MAPK5 or SPL11. So all these guide RNA there are there, and now we have confirmed these 2 construct. We also make we clone this particular gene back one in binary vector. We confirm this binary vector, and here we have started doing rice transformation of this particular gene in Kitake. So here this Kitake, again this is a very useful rice line, which life cycle of this Kitake is just 60 days. So within 60 days, we can get complete seed to seed cycle of this particular line within 60 days. And again, second thing is that this is photoinsensitive. So in laboratory condition, we can get almost 5 to 6 generations in a year from this one. So we are standardizing its, we will say transformation as well as in planta transformation, so that through in planta we can develop transgenics using this one. So this work is also going on in our lab, and I will take 5 minutes more, where we have also developed comprehensive database for rice blast and bacterial blight using all available, we will say, microarray data. So whatever this transcriptome data and microarray data is available online, we club this data, we normalize this data, and further we identified genes related to resistance, genes related to susceptibility, then genes specific to particular strain. Like this we have identified, and we publish this in 4 impact factor journal database, Oxford database. So what we did, we have taken these GEO data sets, and through Limna R code, we normalize this. And after normalization, we check its expression. And further, we develop this rice metastasis database, where we can identify gene on the basis of genotype, on the basis of tissue, on the basis of stage, on the basis of strain, on the basis of days post-infection or hours post-infection, or we can say we also can do common gene search. We can do gene search on the basis of, we also can identify SSR markers related to these genes. Even if someone has identified some QTL, so how many genes are there which are expressing within the range of this particular QTL, those can be also identified, and tolerant into susceptible genes also can be identified using this one. So I will request you all to visit this rice metastasis B database. So in which this is example where this rice blast related genes can be identified for leaf, root, seeds, seedling, or root versus leaf, or even at different days of infection, or through different strains, how it is happening. So again, we can identify these dimers, SSRs on the basis of di, tri, tetra, penta, or hexamers were present there. And we also given one tab where we can identify common genes between bacterial blight and rice blast, so that what are the common genes present along this one. So this is here we will say homepage of this one. And most important thing which I like here, all breeders can be used this particular database. So what we have shown here that how we can utilize it for panicle blast analysis. So basically we have given all IDs of Werke genes here, and we identified genes which are upregulated using all those normal blast expression. Then from our data also we identified the expression of TTEP genes, and we identified these 12 genes which are upregulated in TTEP, and its even expression is slightly upregulated in TTEP, and there are few genes which are not upregulated in HP2216. So from this data, what we did from blast experiment and from our experiment of panicle blast, we identified common 8 genes and 4 these genes, and we took this all 12 Werke genes. We checked those genes for protein variation in this TTEP, HP22 and Nippon bear at gene level expression, at protein level, and we also check it 1 kb promoter of this one, and we identified that this 26, Werke 26, Werke 64, and Werke 76, these genes have different promoter. Again, in this 26 and this 76, these have different gene also. So these genes are probably important, which are present in TTEP line. Again, we similar way here in this case, what we did, along with resistant and susceptible line, we also take 2 strains of magnoporthe, so that there is checkerboard of resistant line and virulent strain, resistant line, a virulent strain, susceptible line, virulent strain, and susceptible line, a virulent strain. And then we check the expression of all these genes, and where we identified there are few genes, like if we say This is 76, 64, and 58, and mostly this 7. So these genes are upregulated only in resistant line as compared to susceptible line. And if we see its expression is very high only with virulent strain. Okay, so it means these are the genes which are really important, probably providing resistance. Okay, so now we are characterizing these genes Werke 7, Werke 58, Werke 62, Werke 64, and Werke 76. So these are only expressed in resistant line against virulent strain. Okay, so now if we talk about there is in database or in anywhere, we will say 3000 rice genome is available and from almost 89 countries. Okay, and this data is available for all rice researchers. And if we see it here from this database, almost 2 crore SNPs are present. So if we try to characterize these genes, we may get this information which is available on these Giga Sciences, and again SNP SIG, where we can get this information. So this particular can be useful for characterizing these genes. So in summary, I will say we did transcriptome of panicle blast and identified almost 3,553 genes in resistant line and in susceptible line, and 2,491 in TTEP line. And we propose that cell wall strengthening is the major mechanism of panicle blast resistance in rice. We also identified novel genes like VWF, having domain of VWF, domon, blupensin, PCAP, then this PMR5 or EAP genes. Then we also identified 6 structurally different genes which we are characterizing. We also identified negative regulators, and we are characterizing this through multiplex CRISPR assay. We evaluated 60 and 22 lines, and out of which we identified 3 lines which are providing resistance at 3 different places, that is Hazaribagh, Delhi, and Meghalaya, and that too for both panicle and lip blast. We identified 2,688 NBS LLR genes from 11 species. We developed this rice MetaSysB database, and through which breeders can identify genes which are present in their QTLs. And we also identified some Werke genes which are really important for this panicle blast. So these are our publications, and whatever work I have shown here, this work I will say is yet to publish, and this is in communication. So along with these papers, that will be again there with me. And finally, this is my last slide. So here, I want to first thank organizer to give me this opportunity to present my work on this panicle blast at this international forum. And along with this, I want to thank Dr. T.R. Sharma sir, who is right now DDG Crop Science in ICR, and who initiated this work, and I am carry forwarding this work on blast. He work on blast, and I have initiated on panicle blast. I also thank our director, then Someshwar Bhagat, G. Prakash, and Uma Kanta, who is helping me to characterize this one. And along with this, my colleague Amita Mitra, all my co-PIs, and obviously my students who are working on field and in lab to characterize these genes. So now we have treasure of genes, and slowly we are characterizing those genes so that we can get better resistance for panicle blast in coming years, and those lines will be available for Indian as well as global farmers. Thank you very much.
Thank you very much, Dr. Solanke. What a wonderful talk, and it is ample amount of work you people have been doing there. It's a great work. So I thank you for giving this wonderful talk on this platform. Sir, can you stop sharing your slide?
Sure, sure.
Yeah,
Thank you.
okay, good. We have some question in both the platform, in the Zoom as well as the YouTube. So I'll come to that in a while. So I have a couple of questions, sir. One is very general question because I am not in of this field. How does this Megnaporte naturally infect the rice? Because you people are using the artificial method by injecting them in the position where you want to study them. So in natural condition, how would they infect?
Yeah, actually, in natural condition, if we see on lip, this particular spore, uh, which fall on this particular lip, and after that, it try to degrade its cuticle layer, and after that, it form aprosorium, and it enter within the cell, and then it propagate there through mycelium. So this is natural one, but in Delhi condition, we were not getting this panicle blast. Although sometimes we get lip blast, but we don't get panicle blast. And again, panicle blast cannot be done under control condition, because we have to grow all those plants till till getting panicle, and then managing those plants in control condition. It is really difficult. So we develop this particular method.
Another small question, sir. Like everybody are talking about the resistant gene present in the resistant line. So, as you have shown, like around 2000 gene were upregulated in your sensitive line.
Exactly.
So, are those genes something of importance, or they are like because maybe I missed, like it is not spoken about in your talk, or are people working on that part of the genes? Why are
Uh,
so much upregulated in the sensitive line if they have no role, or can we target them for some good work for the?
yeah, because that's why to minimize those genes, possibility of identifying those genes, because what we did first, we remove those which are present in control condition, and only those genes we have taken which are upregulated in these time points. And again, what we did, we are comparing this with susceptible lines. So even upregulation of some genes which is present in resistant line, but similar genes are upregulated in susceptible lines. We are also removing those genes, and we are taking only those genes which are upregulated only in resistant lines, or sorry, not upregulated, I will say differentially expressed only in resistant lines. So two-way reduction is there. So we are comparing with control condition, as well as we are removing it by comparing it with susceptible line. So only those genes we are considering which are present only in resistant line and during infection.
Yeah, my point was if they are expressed more, uh, can we target them in the which are in the sensitive line? You can target them so that you can make the line resistant.
Exactly. There are some genes like R genes, if we say, most of the R genes, if we talk about, those are continuously expressing. So those
Okay.
are not upregulated. So I am talking this about PAMP resistance, rather than
Okay.
we will say it another resistance.
Okay.
Okay,
Okay.
ETI resistance. Because in ETI, what is happening? Most of these R genes, these are constantly expressing. So this whatever we are talking about, this particular mechanism doesn't work with R genes. So R genes we cannot identify it using this one. But yes, structurally different R genes, and again those are expressing under this condition. Those we can identify. But there are few R genes like even PI54, which is inducible. So
Okay.
those genes also we can take. But other than this, like transcription factors or kinases, these genes are obviously, if those are upregulated, then only we can select those genes.
Okay, thank you, sir. So now I'll come to the question from our participant in this panel itself. This is Dr. Amrita Panda. It's a technical question, sir. What is the red and green color indicates in differential gene expression data?
Okay, it's up to you. If you are giving red color for upregulated genes, we will say those are upregulated. And green color too means it is depend on us. Sometimes we from yellow to blue also we give. So this is on us.
This is another question, sir, from Anil. He is asking, are 92 lines sufficient for genetic study?
Definitely not. Okay, but since in this case we have almost, I will say, 50K chip. So with this 50K chip, even if we get 20% genes are variable, so we will say we will get 10,000 SNPs we can get. So from those 100 lines or 92 lines, if we are getting, so we can narrow down these things, and further after getting that we can again cross, and in F2 population we can get. And in this case, since we are using real population, so this real population is almost stable, and this is immortal population. So at least to narrow down the reason, we can use these particular lines.
Right. Okay. Another question from Rujita Mishra. Does the spore attack of the pathogen changes carbohydrate cycle? Is photosynthesis hampered? Actually, there are three core questions together. And what is the ROS site, production of ROS site when you have the pathogen attack?
So obviously, when there is infection, that is our hypothesis that probably these all those carbohydrate related genes, or it get activated, or there is a breakdown of some carbohydrates, and because of which signals are formed. So these glucans, already these are known, small glucans of 10 to 20 amino acids. So these are act as a sensors, or those are providing, these are telling that particular cell that is there is something problem is there, and immediately it increase ROS. So when this ROS is all of a sudden increase, it activated other pathways for
Okay.
resistance.
I think Professor Kutubudin have a question. Sir, you can directly ask. You can,
Okay,
yeah.
hello.
Yeah.
Good afternoon, sir.
Hi,
Thank you for your nice talk. So I am from CRRI Katak, Center
hi.
for Research Institute. Yeah. So yeah, it was wonderful to listen to you. But one of the slides when I saw, I was so happy to see that the slide that when you told that you are using Kitake as your transformation material.
Yes.
So I recently got some seeds from Erie, Philippines, from the gene bank, but unfortunately, none of them germinated. So do you have any seed to
Please,
spare?
please, whenever you you want,
Yeah.
yeah, yeah. Because this year I have grown only seed where there was plot just to grow this for transformation, because we are using it for implanta.
Okay.
So for one experiment we need more than 500 seeds.
Okay. Okay.
So whenever you required, I can send you. No
Okay,
issue.
sure. So I would write write it to you.
Yeah,
I worked on Kitake and it was so easy to transform.
yeah, exactly.
So
But we are also trying to develop in planta method,
okay.
so that simply within 3 months we can get transgenic.
Okay.
So that is our idea. So we can grow these seeds, and after germinating these seeds, immediately inject these seeds, and whatever you will get, those will be transgenic. And after 2 months or maybe 3 months, when you will germinate and with any reporter or any control, immediately we can identify whether which is transgenic or not. So that is our idea.
Okay. Thank you very much. So you know.
Yes.
Maybe network issue.
Yeah.
So in the meantime, maybe I can ask the question from YouTube.
Please.
So from YouTube, people are asking, Biswajit Sahu, is it possible to replace the present high yielding susceptible variety with resistance variety with same yield potential?
That's what right now we are doing. Actually, we are not developing new varieties, but we are trying to improve this, like improve Pusa Basmati, if we say, or all other improved, what is this, Samba Masuri. So all these varieties where we are simply incorporating these genes, and through background and foreground selection, so that for foreground selection for this particular gene and background selection for other gene. So there is no need to replace those varieties. It is better to incorporate these genes through pyramiding, and through marker state selection, and keep those best varieties as such, with additional advantage of resistance to this blast and blight etc.
Yeah, one more person asking, what decides a rice to be resistant or susceptible? Which secondary metabolite related genes express high in TETI?
In
TETI.
Okay, this metabolites we haven't checked it. That is, I think.
Some genes, metabolite genes. I think so.
Okay, metabolite related genes he is asking.
Yeah.
Because we haven't checked metabolite, but yeah, there are some secondary metabolites. Right now, I'm not getting its name, but yes, that particular is there.
Okay.
When metabolite is there.
Yeah, Vidya madam.
Yeah, sorry, I think there was some issue, so I was like disconnected. Okay, so there are some more question in the YouTube. Is it possible to replace the present high yielding susceptible variety with the resistant variety with same yield potential?
Yeah, just now I have
Yeah.
answered this one.
YouTube
this
one
is
of that.
done.
Yeah, YouTube I asked.
Oh, sorry. The other, all are done from YouTube, Dr. Vinod?
Yeah. YouTube I did.
Oh, good. Then I think there are no more questions.
There is a sports Bharti something asking.
Actually, my chat
Zoom?
box, the question has
So,
been erased.
So maybe I am telling that one.
Yeah, please go ahead.
So sports Bharti asking, in wheat there are A, B, D genome because C create chromosome breakage. So remove BT the natural selection. Is any this type of situation found in rice also? And in wheat we require 6 to 7 generation for RIL generation. How many generation required for RIL generation in rice?
Pardon, I don't.
Yeah, some background.
Can you mute? Yeah, can you mute this first part? I think there is a noise from that participant.
Yeah, I'm doing that.
Dr. Vinod, can you repeat the question?
Yeah, I'm doing that. So just a minute. So I was muting some people. So in wheat there are A, B, and D genome because C create chromosome breakage. So it is removed by the natural selection. So is there any type like found in the rice?
In rice also there are many genomes, but mostly all those 11 species, only AA genome is there.
Right.
So although D genome is there, F genome
Some
is
wild types.
wild type, but those are not crossable. But still, which are close wild species, all those have AA genome only. Like if we talk about Rupi Pogon or Nivara, those also have AA genome. So there is no issue. Yeah, obviously, since these are different species, there will be cross compatibilities there, but still those are crossable.
And still that question is there. So in wheat we require 6 to 7 generation for RIL generation, but how many in rice is required?
For real, that is natural in any plant. If we or any crop we are generating reels means we have to stabilize those. 7 to 8 generations are always required. Either you can map it in F2 generation or you have to wait for this immortal population of reel.
So one more from YouTube is coming.
We are running out of time also, Dr. Vinod. So
Yeah.
I think last question.
Maybe this will be the last question.
Last, yeah.
So Dorakya asking, sir, as you concluded, cell wall strengthening is provided during the panicle blast. Did you check any cell wall degrading enzyme from MRIG?
We got some like EAP. But we are not yet confirmed. We are characterizing those genes like eukaryotic aspartyl protease. This is the gene which we have identified, but we need to, and till date, we have not initiated work from magnoporthicide.
Right.
So when we will characterize our genes and we observe that yes, those are giving resistance, then we will try to identify mechanism how those are working through magnoporthicide. But definitely this EAP, we are develop its protein and we will check whether it is really degrading magnoporte cell wall or not. That we
Yeah.
are doing. One of my PhD student now is
Yeah.
he started with this particular EAP1 gene.
Yeah, so many questions. So it's like it will keep continuing. Many questions are floating. So please send the questions to us, and we'll be also addressing in the meantime. Okay. So and anytime also you can contact Dr. Amolkumar. So he will be also happy to share.
Definitely.
Yeah. So then,
Okay,
Vidya madam, please.
yeah, I think thank you so much, Dr. Solanke, for giving a wonderful talk. It was like flooded with information, like interesting information. So I hope all the participants have been benefited with the information. So I again thank you from organizing team for your valuable talk. I'm looking forward to have you physically sometime here in the institute. So we have now in this session we have another speaker who already have joined in our panel, Dr. Kutubudin Molla. So he is from NRRI Cuttack. So he will be talking in a while now. So let me introduce him. So Dr. Kutubudin is an agricultural biotechnologist, and he's working at this Cuttack. Sorry. Odisha, and he has published various reputed publication in the reputed journal. And he did his postdoc research in Pennsylvania State University, USA, as a Fulbright Fellow for 2 years. And recently, he has been awarded with the INSA Medal for Young Scientist 2020 by Indian National Science Academy. And Dr. Molla is interested in precise genome editing in rice and uses base editor and HDR method for rice crop improvement. So his group is engaged in developing genome editing tool for plant and utilizes for the crop improvement. So plant pathogen interaction studies use many technology to engineer the crop and generating transgenics, which however are not accepted by a user due to the foreign gene. So latest molecular biology technique delineate the targeted engineering of the crop more specifically. So which he'll be talking about in his this talk, and the title of his talk is replacing one DNA letter with another by CRISPR-Cas tool, which is a very interesting methodology which has been used recently. Everybody is using, and everybody knows the this developer of this technique were awarded with the Nobel Prize. So I welcome Dr. Molla, and we are looking forward to have a wonderful talk followed by the previous one we had. So please, Dr. Molla.
Okay, thank you, Dr. Vidya Devi Negi. And thank you, Dr. Binod Bihari Shahu, for inviting me. It's my real pleasure to be here and to talk about this exciting technology. And rather, what I would say, the conventional CRISPR-Cas9 that most of you are aware of, but I would be talking about a more advanced technology today. And also, thank you for another panelist, my previous speaker, Dr. Amolkumar Solanke. It was a very wonderful talk that I listened to him. And okay, so no need to waste more time. Directly going to this technology, that technology will allow us to change or replace one DNA letter with another letter that we want to change. It's not a random change, but it's a precise change that we will be talking about a lot today. So if I see this, if we see this paper that came first in the Science Journal in 2012, June 28, and that paper was communicated by Jennifer Doudna and Emmanuel Charpentier. And that paper actually took the whole biological world by storm, And in the Nobel history, this is the first time that it took only 8 years from the first publication to a Nobel Prize. So this year, the Nobel Prize has been awarded to Emmanuel Charpentier and Jennifer Doudna for their discovery or development of a method for genome editing. And that technology is nothing other than that CRISPR-Cas9 technology. So I will be talking a lot about this technology today, not only limited with this conventional knockout by CRISPR-Cas9, but going beyond of this technology. That technology is called base editing. So yeah, let me come to my outline of today's presentation. In the first part, I will be introducing you to briefly mentioning about what is genome editing and how CRISPR-Cas9 is actually utilized for genome editing. And then I would be moving forward towards more precise one that is called base editing. And then what are the platforms available for performing different kind of base editing. And in part 2, I'd be giving you a glimpse of my research work related to base editing in RISE. And then in part 3, I would be take you to the major limitations of this technology. And in part 4, I'd be just briefly mentioning what are the potential application of this technology and why is base editing coming in a big way in the recent times. So, okay. So it's always good to have rephrased about our understanding of DNA and gene in order to smoothly follow my presentation. You know, any higher eukaryotes like human is made up of trillion of cells, and like also a rice plant is also a higher eukaryotes. It is also made up of trillion of cells. So if you see, this is an example of plant cell, and this cell has a nucleus. This nucleus is a very important organelle of this cell because it contains the genetic material that is called chromosome. And this chromosome is nothing but a long thread of DNA that is coiled around a protein. That protein is called histone protein. So forget about that histone protein, but just remember that chromosome is made up of long thread of DNA that is coiled around each other. Okay, so this DNA is our genetic material, but the whole range of DNA, the whole length of DNA are not equally active or equally important for a cell according to present understanding. You know, so a functional portion of that DNA is called a gene. A functional unit of that DNA is called a gene. So because this portion of this functional unit actually governs a character by means of its expression. So how a gene expresses, it is nothing but, you know, gene means a part of DNA which is functional, and that DNA produces RNA, and that RNA produces protein. So this is called central dogma. So DNA to RNA and then protein, and that protein controls a character or a phenotype by acting in a biochemical pathway. So it means one gene or maybe several genes controls a character. Okay, so what is there in our gene? If you see, our DNA has 4 different letters. It is nothing but a different kind of sequences of 4 letters, that is adenine, guanine, cytosine, and thymine, that is abbreviated as A, G, C, and T. And different sequences, they make up the genetic code, and this genetic code actually controls almost everything in an organism. Okay, so DNA is nothing but different kind of sequences of these 4 letters. Now, as this webinar is on host pathogen interaction, so I decided to introduce you to the genome editing by this classical example of a plant pathogen interaction system, like, you know, Xanthomonas oryzae that causes bacterial blight of rice. This is a bacterial pathogen. So if you see this example of a rice cell, this is a rice cell, and it has certain gene. That gene is called sweet gene. This sweet gene produces a protein that's called, this is the protein and this is the gene. So this protein actually is a transporter protein. So it has a function. It transports sucrose from inside the cell to the apoplastic region, to the outside of the cell. Okay, so this is simple rice. Now, whenever there is an infection from this bacteria zoo or Xanthomonas oryzae, then it secretes some protein, that protein called TAL effector, transcription activator like effector, is a protein or effector protein that goes inside the nucleus and bind with the promoter sequence of that sweet gene. Now, what happens when this TAL effector binds with the promoter sequence of that gene? Then it actually induces, this binding induces the expression of that sweet gene, so that the more number of sweet protein produces. Okay, so here you can see from this schematic that there are a lot of sweet transporter protein is present in the cell membrane. So now, as a result of this more expression of that protein or more production of this protein, then more amount of sucrose would be transported to the apoplast. Okay, so now, you know, zoo actually, this bacteria, they enjoy sucrose as their nutrition, okay, as their food. So whenever there is more amount of the sucrose in the apoplastic region, so it is available to them as a nutrition. Okay, so they will also divide more because they have ample amount of this is sucrose or this nutrition. So they will grow more and they will infect more. So this is, you can say, this is a classical example how a bacterial pathogen uses plant's own gene for their benefit. So this gene, if you see in details of this gene, a little bit zoomed view. So this region where the tail effector binds is called effector binding element. This is not that every where tail effector can bind in every portion of the promoter. It's not. There is a specific somewhat around 20 to 24 base pair in the promoter region where specifically the effector protein binds. So this is called effector binding element, and because of this binding, the sweet expresses more, and that helps the bacteria, not the plant. Now the question here: can we specifically disrupt the EBE sequence in the promoter? So this is a simple hypothesis you could think of, that if we could disrupt the sequence of EBE, not disrupting the sweet gene, but disrupting the EBE sequence, then we could inhibit the binding of the tail effector, because this binding is very specific, as I mentioned earlier. So now if you change the sequence of EBE, by any means, you could inhibit the binding. And whenever effector cannot bind with the EBE, then this bacteria would not be able to use the sweet gene to produce or to transport more sucrose for their benefit. Okay, so in order to answer this question, the way is genome editing. Yes, you could specifically change a target region sequence by this method. Is called genome editing. So it's kind of, so I told you that DNA is nothing but a complete sequence of DNA letter. So if you change that letter, and that is called genome editing. So you have a draft ready, suppose, and you want to get it checked by your SAR or by your supervisor, by your teacher. So you give them that draft, and they will find some problem and they will change this word is not good, replace with another word like that. So that is editing of your manuscript. Okay, now this is editing of your target region. So suppose this region is the sweet promoter EBE. Now you have to block that from binding the effector region. So change the sequence in such a way that it no longer works for the bacteria. Okay, so that is genome editing. Now, how it could be done by genome editing? So there is a lot of few tools of genome editing like TALEN, like ZFN, and then this CRISPR-Cas9 that I am talking about today. But the CRISPR-Cas9 became more popular because it is efficient, it is very simple to use, and it could be easily replicated, and it is cheap also. So if we see CRISPR-Cas9 is like a Caesar, is kind of like a Caesar. So how it actually works? This CRISPR-Cas9 has two components. If I want to give you a very simple example, so it has two components. One is a customizable RNA that we call as a single guide RNA. Another is a DNA cutting protein that is called Cas9 protein. So if I want to give you another simplified version of analogy, then this Cas9 is like a postman that has two scissors in its pocket. And this one is like a genomic address. This is an address and this is the postman. So if we give this address to the postman, postman takes the address and it goes to the genome to find the similar address. It means the RNA bases, it searches for the similar sequences in the genome. Whenever it finds exact similarity with this RNA, then it binds to a similar DNA sequences, and when it completely binds, then this postman gets the signal that this is the region where I need to make a cut. Okay, so now this Cas9, as I told you, this postman has two scissors, then it would make a double strand break in the DNA. As we know that DNA is a double strand structure. So one scissor cuts one strand, another scissor cuts another strand. So this is how CRISPR-Cas9 could be used to make a double strand break in any of your target gene. Okay. So now the question is how it actually mutate or how it actually edit the sequence. Yes, it is clear that it can target it in a targeted fashion. It can make a double strand break in a region where we want. But how it makes? So how it makes the mutation? So the answer lies in the cellular repair pathway. So DNA is a very important material. As I told you, DNA is the genetic material which is very crucial for a cell survival. So once the DNA is broken, cell takes it very seriously. They don't sit idle, but they immediately activate its own repair system to religate that broken end. And there are two repair pathway in the higher eukaryotes. One is non-homologous end joining, and that's the predominant pathway. Most of the time, cell repairs their DNA break with this pathway, but this pathway is an error-prone pathway. During these repair system, due to error, it inserts or deletes some original bases. Suppose here you can see this red one is actually designates the insertion of new bases that was not present in the original gene sequence. And similarly, some original sequences, one or two bases or three or four or five nucleotides could be deleted from that original sequence. So once new insertion happens or new deletion happens, then the gene sequence is disrupted. So the sequence is gone. So the open reading frame of that gene is disrupted. So this gene cannot produce the same protein as it was producing in its original form. So now the gene is mutated. So the gene function is gone. So this is a very, very efficient pathway, efficient method that a lot of people uses this one or harness this one for generating a lot of mutations. Okay, so as you heard from Dr. Solanke, that he was also using to mutate the genes by using this pathway. Okay, so there is another pathway, it's called homology directed repair, but this is a very, very low efficiency pathway in higher eukaryotes, especially in plant cells. So this does not happen always, but this happens in a particular portion of cell cycle. So if you see how it happens, in normal cases, in a diploid organism like rice, there are, two chromosomes. I mean, two sister chromatids. There are two copies of each chromosome. Okay, so if one copy of that DNA is broken, then it looks for the another copy which is unbroken, and it copies that sequence, the unbroken sequence, to the broken one to repair it precisely and near perfectly. Okay, so this is called homology directed repair. But if you want it for your genome editing purpose, you have to supply an artificial donor template that has also it looks like similar with this sequence. You can see this green portion are all similar with this DNA, left arm, right arm, but you have to introduce something in the middle, the change that you want to make. Now, if you supply inadequate in a large amount to the cell, and you make a double strand break, then cell will take this donor template that you artificially supplied as a sister chromatid, and you are tricking the cell to repair the break by seeing the donor template, but not seeing the sister chromatid. Okay, so if they copy the sequences that you presented through donor template, then your intended change is also copied to the genome. So now you get a precise editing. Okay, but this is extremely low in efficiency, as we also tried a lot of times. Okay, so now this video will make you more clear about how actually this CRISPR-Cas9 technology disrupt a gene function.
There are two components to the system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome, PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, fixing process is error-prone and often inadvertently introduces mutations that disable the gene, This makes CRISPR a great tool for knocking out specific genes.
Okay, so if you see, I think it is now more clear to you. So coming to the same story that I told you about the sweet one. So this sweet actually is a susceptibility gene that you heard from also from Solanke. He talked about in one of his slide that susceptibility genes that actually help in early pathogen establishment. This gene is present in host plant, but they help pathogen for the establishment. And it also helps in modulating the host defenses, means it reduces the host defense. And then it helps in pathogen sustenance, like this sweet one helps by transporting sugar for their benefit, for their bacterial benefit. So, you know, just to give you example, this sweet gene has been disrupted by using several genome editing technologies like TALEN, CRISPR-Cas, and all of those earlier reports that showed very effective resistance when this sweet gene promoter, this EBE, has been disrupted. Okay, but the gene has not been disrupted. The promoter sequence, the EBE sequence, has been disrupted, and you can see this panel is the non-edited version of that plant, but these are all edited version. It means those plants has the mutated EBE sequence in the promoter of sweet genes, and they are incompatible incompatible for the bacteria. So bacteria cannot make severe infection as they do for the wild type or control plants. Okay, so this is a classical example how genome editing could be used to develop disease-resistant plant by mutating susceptibility gene promoter. Okay, so now, coming to the next part of my presentation. So this is a snapshot from a famous Indian movie. If I ask you, I think most of you could remember. This movie is from Pa movie, and this is Amitabh Bachchan. He was acting as a Oro, and this is Oro's friend. They are both are 12 years old. And if you see Oro, by seeing his face and his body, he looks like a very aged person, okay? Because he is suffering from a rare genetic disease that's called progeria. And this progeria causes rapid aging at the childhood of an infected child, okay? And they look like a very aged person. What is the difference between these two? This healthy girl has a G in a gene, in a certain gene. That gene is called lamin. Whether the Oro has an A nucleotide in the same position. So because of the single nucleotide change, of the single DNA letter change, Oro has died at 13 years of his age. So we knew the reason of his suffering because of this genetic, one single wrong genetic letter, but we did not have any answer to cure that. Similar example, if you can see here, this is a rice panel Kitake, as told, and we interacted about this rice variety. This Kitake is susceptible to a blast pathogen, but here this mutant is tolerant to that pathogen because of the single nucleotide change. Similarly, it is present C in the BSRK gene, but T is present instead of C, and because of this T, this mutant become resistant to that disease. Okay, so this is the plant perspective. So the question is, okay, is there a way to rewrite genetic letters? So we have to specifically rewrite from C to T and here G to A. So in order to cure them or to develop a resistant plant, but non-conventional CRISPR-Cas9 cannot do. They can disrupt a gene, but cannot precisely correct a gene. Okay. Now, okay, so if you want to go for correction of that kind of single change, then you have to go through the homology-directed repair pathway. But that is, I told you, extremely low, very, very low in efficiency. So that's why it is very difficult to achieve this kind of changes in the genome. And also HDR has a lot of other problems, like as I told you, it is innate low efficiency and high indel. It could also generate high number of insertion and deletions. So besides correcting the gene, it can also disrupt the gene. So the condition would be more worse. So also it is cell cycle dependent because this HDR is active only in AS and G2 phase of the cell cycle. but it is very difficult to restrict your cell to this space and perform the HDL editing. So it's very difficult. Then also difficulties in donor template delivery. As I told you, for performing HDL, you have to supply an artificial donor containing the intended change at the middle of that donor, but supplying an additional donor in adequate amount is also very difficult. Okay, so that's why this technology is called base editing, which is you could call it as a pencil. If you call CRISPR-Cas9, conventional CRISPR-Cas9 as scissor, this should be called as pencil because it could rewrite from one DNA letter to another. Like it could go in both directions, A to G, both directions from T to C, and also recently developed C to G. So it is nothing but a fusion of N-Cas9 with another protein is called deaminase. This N-Cas9 goes and binds with the DNA, target DNA, and this deaminase actually do the job of rewriting the letter. So what is N-Cas9 that I have not introduced you to? So Cas9, I told you Cas9 is like a postman that has two scissors, but this N-Cas9, if you take out or snatch one scissor from that postman, so it becomes, it has only one scissor left. And this Cas9 is called Niki's Cas9. So it is able to make a single strand break, not a double strand break, because it does not have two scissors, it has only one scissor, okay? So now this N-Cas9 has been used for base editing development, base editor development. So this is a small video to make you more clear.
Then to transport those enzymes to specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you can turn a disease-causing mutation to a healthy version of the gene.
Okay, so now coming to the platforms, different platforms what are available for base editing. So these platform is called cytosine base editor that could be used to alter a cytosine to a thymine in one strand. Now opposite strand, if you see, it could be also used as G2A change. So it is nothing but N-Cas9 fused with cytidine deaminase. That is, this is R-ApoB-C1. This is taken from rat. That's why it's called R-ApoB-C1. This is another platform, same N-Cas9 is fused with another cytidine deaminase from Petromycin marinus, that is C. lamprey, and fused with another protein that's UGI, uracil glycosylase inhibitor. So these three proteins together could achieve a targeted C to T change in the genome. So coming to another one, that is adenine base editor, ABE. So these both the base editor platform actually pioneered by this David Liu from MIT Harvard. And so this adenine base editor is developed by fusing N-Cas9, that is Niki's Cas9, with a dimer of adenine deaminase. This is E. coli tRNA adenosine deaminase. But you remember, this is a tRNA adenosine deaminase. So these deaminase can deaminate an RNA, but not a DNA. But what they have done, this David Liu's group amazingly, that they have artificially evolved that RNA adenosine deaminase to act on DNA substrate. So they evolved from an RNA deaminase to a DNA deaminase. Okay, and then they fused with the N-Cas9, and it could perform specifically A to G change, A to G conversion, and in the opposite stand T to C conversion. But it has this kind of optimum window. Suppose this is the target region in your genome, this 20 base pair that we commonly call a spacer sequence in the genome. You have to select for any genome editing experiment this 20 base pair, and this is called PAM. PAM is protospacer adjacent motif. So your target region should be followed by another 3 base, that is NGG, and that is specific for this kind of Cas9 to bind. Otherwise, it cannot bind, especially for SP Cas9, Streptococcus pyogenes Cas9. But if you take Cas9 from other bacteria, it could bind, it could recognize other PAM sequence, but not this one. So what I was trying to make a sense here, that your target adenine, if you want to change an adenine to G, then this adenine should be present between 4 to 8 base of your spacer sequence. So this is an optimum activity window for this base editor. But another version has a little bit extended window, like 4 to 10 base. But if you have, suppose here at the 17th number of base, and that you want to change, then it is very difficult to change that using this platform. Okay, so that's called activity window. This is called optimum activity window of base editor. Okay, now how this adenine base editor actually works to correct an AT, suppose A to G. Suppose this is an AT base pair that is a defective one, and you want to correct them to a CG base pair. Okay, so once we apply ABE adenine base editor, so this adenine base editor has this deaminase enzyme. So what it would do, this deaminase would deaminate the adenine to make it a inosine. So now it becomes inosine and T, so IT base pair. But IT base pair is a mismatch. It is not a common base pair. So then cell's repair machinery is very active. So they always sense this mismatch in the genome, and they come here to repair. But they are confused which one is the mismatch, whether to remove I or whether to remove T. Then the Ncas9 actually works here. This is a very smart designing that Ncas9 makes a special cut or single cut in the T containing strand, not the I containing strand. So once the T containing strand is cut or broken, then the cellular mismatch repair machinery recognize this strand as the wrong strand because it is already broken. So now what they would do, they would remove the T and it would incorporate a C opposite to I, because always opposite to I, there is the more tendency of incorporating C. So now this IC base pair, now in the next phase of replication and repair, it would be corrected as GC base pair. Now you got your expected result AT to GC, and this is the pathway. Okay, so now coming to my part 2 of my, so I give you some practical example how it could be done and what the results look like. So here, what we have targeted this gene, white stripe leaf 5. These mutant phenotypes actually develops a white stripe leaves in the early developmental stage of rice plant. So this is because of A to G mutation and in the opposite strand T to C. So here is the target. This is a snapshot of that gene sequence. So when this target A is changed to a G, then it should produce a white stripe leaf phenotype. Okay, so that was our target to develop this kind of mutation with base edition. Okay, next another gene we targeted also, that is called zebra3, and that develops a dark green sectors and mature leaves, like this is the light green, dark green, light green, dark green sector. So that kind of phenotype also comes from another mutation like A to G. This A is the target A. When this target is mutated, then it will develop this kind of mutant phenotype. Now, if you see this one in details, that this target A falls within the recognition site of a restriction enzyme. So this is the recognition site GTC-GAC. So once this has been changed or mutated successfully, so the recognition sequence of that restriction enzyme is changed or altered. So if we get successful editing, then the SAL1 enzyme cannot make a cut in that gene sequence. Okay, so that was just to describe, because my next slide actually related to this one. So okay, so what we have done, as you briefly mentioned by Dr. Solanke, this is called PTG system, polycystic tRNA gRNA system. So so this is developed from Young Lab. So I was working with Young's lab, and one of our earlier postdoc, Kevin Z, he has developed this wonderful technology, and that is being followed worldwide. That's called PTG, or polycystic tRNA gRNA, for multiplex gene editing. So multiplex means in one go you could target more than one gene. Okay, so here our target is 2. One is WSL5, another is CEF3. So what we have done, this is a guide RNA for the editing WSL5 until this portion. But we have fused a tRNA at the 5' end of this guide Also, another tRNA at the 5' end of another guide, and they are fused together. They are transcribed by a single promoter. So they look like this one: tRNA, then guide RNA, tRNA, and then guide RNA. Now, within the cell, plant has two different enzymes, that is called RNase-P and RNase-Z, and they have the inherent capability to cut out the tRNA. So they make a cut here and to release the tRNA. They make a cut here and they release the tRNA. So once the tRNA is spliced out by this endogenous activity of endogenous enzyme, then the guide for WSL5 becomes free, and it would take the base editor and go to chromosome 4, because chromosome 4 is the target for WSL5 where the gene is present. And another Z3 would go to chromosome 3. So they are separated. They take the protein that is in Cas9 and the deaminase, and then they go to their respective target. So that is how we achieve the multiplex editing. So when we did it transient as in protoplast, so what we have done, we harvested the DNA after treating the protoplast with the base editor construct, and after 2 to 3 days, when we harvested DNA and amplified the region of WSL5 and Z3 and digested with SAC1 and SAL1 respectively, and you can see this is the wild type is completely digested. Wild type means it is not treated with base editor, but the other samples are treated with base editor, and you can see here clear undigested bands. So few of the population are undigested, means they are resistant to SAC1 enzyme. It means this is a clear indication that the sequence has been edited, the target A has been edited. So the recognition sequence has been changed, so SAC1 cannot make a cut. Similarly, for SAL1, for the second target Z3, also we have found quite a good amount of resistant band. That's an indication that successful editing at the target loci. So this was our efficiency, where we have found WSL5 is more efficient than the Z3. But from this result, we are encouraged to see whether we could mimic the same kind of result in a regenerative plant. So we designed two different kind of base editor, one is 7.10 version and another 7.9 version. As I briefly mentioned, that this two has a different activity window. This has window 4 to 8 and this is window 4 to 10. So we tried both of them, and then amazingly we have seen in T0 or T0 state, T0 line, this is a chromatogram from a sequencing chromatogram. This is a Sanger sequence chromatogram. So if you see, if I want you to understand very easily, this blue peak means C nucleotide, green peak means A nucleotide, and black peak means G nucleotide, and red peak means T nucleotide. So this is a wild type of control chromatogram of the target region that we targeted. And this A was our target to change. This A was our target to change. And you can see in T0, we have found this A has been changed to a G. But remember, and if you look at very sincerely, you can see here two peak. One is green peak, another is black peak. It means the green peak has not completely converted to a black peak. It means, you know, there are two. I told you that in a diploid organism there are two copies of each gene. So it means this is a case of monoallelic editing. So one copy has been edited to guanine, adenine to guanine. Another copy has not edited. So that's why we got two peak here. But if you match the other peaks with the one, the lower one, we can see all are similar. So it has changed only the targeted, not the adjacent bases. Okay. This is for WSL5. Similarly, for Z3 gene, also this A is our target, and we have found similar result that monoallelic editing, that A has been changed in one of the copy of the gene into a G nucleotide. So we generated second generation of T1. So in T0, when we calculated the editing efficiency, as contrary to our result what we obtained for protoplast assay, Z3 showed more efficiency in the regenerative plants, but W5 showed extremely low efficiency. So now, when you generate T1, means next generation, by selfing the same rice plant, what you can see here, this is interesting to see that this A has completely been changed to a G nucleotide. So this is a biallelic editing. So this is a homozygous or biallelic editing of A to G by the treatment of this base editor. And once we got this homozygous or biallelic editing, we have found those plants that has this kind of editing all showed the expected mutant phenotype, that is white striped leaf. So it means the single change in that gene, as precise changes by base editor, also develop the same phenotype as the natural mutant. Okay. So similarly for Z3 also, you can see this A has been completely converted to G. And we have found this dark green, light green, dark green, light green phenotype along with other expected phenotype. So base editor could able to generate two different kind of mutants, targeted mutants, single base change or alteration in two different rise genes in a single experiment. So we rapidly generated base editor free or non-transgenic mutant obtained in T1. So this is another question a lot of you might have in your mind, that how this genome editing or this base editing technology can generate non-GM or non-transgenic plants. So, if I want to give an example, so suppose when we transformed our plant with base editor construct or DNA construct, then it is integrated into chromosome 1. And now my target is in chromosome 4. So suppose this, once the base editor is integrated in chromosome 1, it produces base editor protein and guide RNA. Then it goes to chromosome 4. Then it changes the target. So it mutated or base edited the target. And now once the target is mutated, now you no longer need the base editor construct to be present in your genome because the work is done. So base editor does not have to be present there. So in the next generation, by virtue of Mendelian segregation, we could segregate out the base editor, and you could select only the mutant plants that does not have the base editor construct. Okay, so this is a snapshot of a representative jellyland. So we selected 25 mutant plants, all are mutated successfully. And when we scanned or screened for the presence of base editor, so we have seen 6 of the plants that does not have this band, specific PCR band, does not contain the base editor construct in their genome. So these 6 plants are mutant, but they do not have the base editor construct inserted into the genome. So now those 6 plants could be called as a non-transgenic but mutant, targeted mutant. So you could obtain very, very easily and very rapidly non-transgenic mutant. Now we move forward to go for economically important traits. So this is a classical example. So we try to correct a premature stop codon. So in a genome sequence, if you have a TAG sequence, this TAG is a stop codon. There are 3 stop codons. So this is one of the stop codons. So the TAG is like a red light to the translating machinery or translation machinery. Whenever there is a TAG, so translation machinery stops there because this is a stop signal. Okay, so now it can no longer produce protein beyond this point. Okay, so because of a premature stop codon, so these variety or genotype is become disease susceptible because it cannot produce the whole length of protein. So what we tried, we targeted this one, and we tried if we could change this A base to a G base, then it would be TGG, and this TGG codes for tryptophan amino acid. So the stop codon is mutated, or a stop codon is corrected to a tryptophan codon. So what we did, we applied with base editor, we could achieve a TAG correction to a TGG. So we corrected a premature stop codon to a tryptophan codon, and now you can see now the translation machinery would not stop here. It is a green signal. So now the translation machinery go beyond this point, and it would produce the whole length of protein, and the plant would be disease resistant. So this is ongoing work. It's not been published yet, but this is to just give you an example how beautiful this technology is. Now this is another recent development. Recently, there is a new class of base editor that I told you previously. The CBE is cytosine base editor. Another is adenine base editor that could make A to G. So if you could ponder on that to both the base editor like CBE and ABE, they can perform purine to purine, like adenine to guanine, purine to purine, and then cytosine to thymine, that is pyrimidine to pyrimidine. So both the transition mutation. but there was no transversion mutation. But recently there are 3 reports that reported that in mammalian cells that there is possible of developing transversion base editor, that is C2G, cytosine to guanine base editor in mammalian cell. These are the 3 platform that has recently been published, and we have just a spotlight or highlighted this article for Science and Genetics recently. You could go through this article in order to learn more how it actually changes SC2G. This is a simple pathway. I'm not going into details. So now part 3, what are the practical limitations of base editors and how they are being addressed? So as I told you that SP-Cas9 or Streptococcus pyogenes Cas9 needs this PAM or protospacer adjacent motif, that is called NGG. So these regions should be present always after the spacer sequence, after the target sequence in the genome. The 20 base pair target sequence should be followed by NGG. Otherwise, you cannot target that region. That is a limitation of SP-Cas9. But SP-Cas9 is the most popular Cas9 till now, and that has been actually initially narrated by Jennifer Dodd and Emmanuel Charpentier in that initial paper, and that is very active. So, but what if there is what if you have a target, but you don't have an NGG PAM in your target region? So what would you do? So there are a lot of other Cas9 orthologs discovered from other bacteria, also engineered Cas9 variant that has different PAM requirements. Like if your target has only NG, it will target by this 2 Cas9. If you have triple TB, then you have to choose this Cas9, this Cas12A. So a lot of base editor also developed on the basis based on those orthologous Cas9 variants, and you could choose them from the toolbox to according to your requirement. Okay. So now another limitation is activity window. So okay, what if you have your target at the 12th position? What if you have your target at the 15th position? So this is called limitation of activity window. So now this base editor, there is A3AB. E3 has been developed that has long extended window that expands from 3rd base to 16th base of the spacer sequence. Similarly, if someone has a problem with longer window, then it has a restricted window, only 3 base, 5 to 7. Similarly, these are other versions of base editor that has extended and limited base activity window also. But you have to choose from the available base editor toolbox. Now, this is another problem with base. This is an example from our study. So this is called bystander editing. What is bystander editing? Suppose this is an example from Z3 target region. So we targeted this A to change to a G. Okay, so we successfully converted it to G. But what if there is an adjacent A is also present? And if that A, you don't want to change that, but that is also changed due to your base editing treatment, then that is called bystander editing. Okay, so we have found few of our plants, not very few, 4 of our plants out of 300 plants. Only 4 plants contain this bystander editing. So the 12th base A has also been edited. So this is we have found. So this is called bystander editing. And then another example, A is completely changed to G. So this is also a bystander editing. And this kind of change also could make amino acid change, and that could also maybe undesired. You don't want that change. Okay. So this is another problem. So this problem has been recently resolved by from Keith Zong's group from MIT. What they have done, you can see this is bystander editing, and this is the cognate that you want. So what they have done, you can see this is a version, engineered version of the base editor that is low bystander editing but high cognate or target editing. So if you have a motif like GCT, this C would be edited. GCT, CG, this C would be edited in high amount, but this C would not be edited if you use this engineered version of base editor. Okay, so now this is the last part of my presentation, and so what are the potential applications of this base editing? So you would get those points from this presentation. So now If you see, as I told you, this is a CBE, cytosine base editor, that has three protein. One is Cas9, another is cytidine deaminase, another is uracil glycosylase inhibitor. Now, if you remove that uracil glycosylase inhibitor, then these remaining base editor can make these kind of changes. It's not only C2T, but it can also make C2G, C2A kind of changes. Okay, so it can make a diverse kind of edit, and it has a lot of applications. Like it could generate molecular diversity, it could have application in directed protein evolution and other also applications. Okay, so this is, as I told you, from my one of my experiment, that it could be used to correct a stop codon or create a stop codon. If you want to create a stop codon, then you have to apply cytosine base editor. These are the three stop codons. Now, if you want to correct that stop codon, as I gave you an example, ABE or adenine base editor could be used to correct them. Okay. Now, another interesting application of this one is, this is an eukaryotic splicing story. So this is an intron and this is an exon. So intron exon has a junction, and the junction has specific bases. All eukaryotic intron has should start with GT and ends with AG. Okay, this is called GUAG rule. So this has a definite role in splicing. So this, if you apply base editor to mutate this donor, this is called a splice donor sequence, this is a splice acceptor sequence. If you mutate them with either CBE or EBE, then you could generate exon skipping, alternative splicing, and intron retention. So you could play with the splicing machinery of the cells with the base editor. Now, as I mentioned here, so it is a great tool for gain and loss of function at single base resolution, and it could generate almost all missense mutation in the cell, and it is a great tool for functional genomics. So this is a classical example for disease application. So if suppose progeria, as I gave you an example, progeria is caused by a single nucleotide mutation. So it was very difficult to generate a model, suppose rapid model, or maybe a guinea pig model or a rat model having progeria disease. So all the disease, genetic disease, they are being studied in a model and not in an affected plant. Okay? So you have to generate or mimic a affected model system to study better that disease. Okay? So it was very difficult to generate models for genetic disease due to point mutation. But this base editor actually enabled us tremendously to develop a lot of single nucleotide, single pathogenic mutation related disease model. Okay, and whenever this model are available with us, so there are a lot of development. I'm not going into details. You have the now you have the chance to investigate that disease better, and then you could strategize any therapeutic strategy by analyzing them. Okay? And now similarly, once you develop a strategy how to treat that disease, then this same editor, base editor, also could be applicable throughout the life history of human, like isolated cell to embryo to newborn fetus to non-dividing cell, even it is applicable to all stages. So the genetic disease having a pathogenic mutation could also be corrected with this technology. So you'd be amazed to know that almost this 47% plus 14% plus 11%, this whole region, this whole percent is like 63, like 72% of the pathogenic mutation could be attempted to cure with the base editing tools available now in our hand. So it is really giving hope to for therapeutic applications. So, these are the few examples that has already been proved or demonstrated in cell lines or disease model to be curable with the application of base editor. Even a recent paper came to cure a progeria diseased mouse with the application of base editor. Okay, and these are all other genetic disease that does not have any answer previously before the base editor came. So similarly, it has tremendous application in agriculture, as it was saying by Dr. Solanke. That suppose he was talking about wild rice. Suppose one wild rice has a different genome, and that is not crossable with the rice, but that wild rice has a definite and a very good allele. Suppose Protetia quaterta is a wild rice species, and that is very highly saline resistant. Okay, so Protetia has a similar gene in cultivated rice, suppose, but Protetia allele has one single nucleotide mutation, and that allele you cannot transfer through crossing, okay, because it is not crossable. Now, what could be done to harness that beneficial allele? So base editor could be an answer to mimic that wild rice allele in the cultivated rice to generate that kind of character or traits. So this is not related, but just let me tell you, this is a fascinating area. This is also, we started working on this one. So suppose there are a lot of people working on CRISPR-Cas9, so we should have a lot of big amount of data worldwide generated. But as I told you, conventional CRISPR-Cas9, now go back to conventional CRISPR-Cas9, forget about the base editing. Conventional CRISPR-Cas9, after it makes a double strand break, it produces random indel. So you cannot control what kind of insertion, what kind of deletion is going to happen in your mutant plants. Okay, so that cannot be predicted also. But what we hypothesized, and based on some earlier studies, that if we have big repair data and we could train a model by artificial intelligence, and we could develop predictor model, and that could predict what kind of guide RNA could give you more mutation. And obviously another option is that what kind of mutation they are going to generate. So the best guide to choose from, and also what guide will give what kind of mutation. Suppose for an example, simple example, suppose this is a 20 base pair spacer and your fourth base is a T. So if you use Cas9, then it would likely give you the most number of mutations that another T would be inserted. It is the most likely outcome of that one. Okay. Similarly, if you have two double C, then one C would be deleted. So this is the kind of prediction you could use for better designing your experiment. So if you want to read more, so you have to read this article. We recently wrote for TensorFlow. Okay, so this is last slide of my presentation. So what we are working now related to base editing is we are targeting economically relevant fine mutations in elite rice cultivars to develop rice varieties. And also we are trying to develop a mini ABE system, adenine base editor, by using only TAD1 monomer and deaminase engineering to generate mobile base editor. Coming to the conclusion slide, what I have told in part 1 and 2, that base editing has unprecedented ability to install precise point mutation without double strand break and without following the homology direction. So nobody has thought about this one. It has been proved effective in animal, plant, and microbes. So part 3, diverse base editing platforms, as I told you, give you an example, are being developed to address the limitations, specificity, PAM compatibility, editing window length, bystander editing, and off-target also. And part 4, during that applications part is basic research, medicine, and agriculture. It is applicable in all the three arena. And it holds great promise for treating genetic disease and also for developing superior agricultural crops. So if you want to know more about base editing, you could go and read my AdGene blog that I wrote for AdGene. It is in a very layman language we have to write. So I wrote this one, it would go through. This is a recently published methods book. So, you know, listening to this kind of lecture is very easy and it's fascinating, but applying that technology to your lab might be difficult if you don't have a good protocol. So this method book actually catalog all the protocols. A lot of experts wrote or contributed for this book and to follow step-by-step protocol. And this is a little bit in-depth review that you could also follow to know about base editing and their possible and potential applications, and also could go through this one. So I want to acknowledge this Professor Young. So I worked there on this base editing stuff on genomic technology, and these are the grad students that worked with me, and I used funding from Fulbright, NSA, ICR, and NRRI. And thank you very much for inviting me. It's back to you.
Okay, thank you very much, Dr. Molla, for giving this wonderful talk and giving a detailed insight of this technology, which, like, at least I was not aware of such a detailed information about it. Thank you so much. So I think there are some questions. Okay, I have one small technical question actually. These
Yeah.
CRISPR-Cas system, they generally target the DNA. Uh, how about RNA being a target for this system?
So are you asking for, is it applicable to target RNA?
Right.
Yeah. So yeah, so this is only 40 minutes presentation, and CRISPR is a skyrocketing technology. So I cannot cover all of those things. I especially covered only base editing, but there is a protein that called Cas13. That's a brother protein of Cas9. So that's an orthologous protein called Cas13. This Cas13 does not bind with DNA, but it binds with RNA and makes a cut in the RNA. So Cas13 could be used for RNA targeting. And in fact, Cas13 has been used recently for COVID-19 detection. This is another area, CRISPR diagnostic.
Okay.
So Cas13 is being used in USA to detect the presence of COVID-19 RNA inside human samples. Okay.
Okay, thank you. Can you stop sharing your slides? So we have
Yeah,
some
sure.
couple of questions actually. One
Mhm.
is from our panel only. This is Rojita Mishra. In genome editing for development of novel trait by CRISPR-Cas is cheaper and sustainable for agriculture practices.
Or genome editing, novel trace. Yes, it is cheaper. Yes, it is cheaper. As I gave you example of how I could achieve, means we could achieve mutant plants, non-transgenic within a single generation. And yes, it is very cheap, and that's why only new thing you have to do, and you have to only change the address. That is the guide RNA. If you change guide RNA, you don't have to change the Cas9 or the base editor. Only change the guide RNA. That would be targeting to your gene, and you are ready to go for your experiment. It's not that you have to design the whole new Cas9, whole new construct. Only the 20 base pair you have to change, and synthesizing 20 base pair, ordering a primer, from IDT. It's very easy.
So that is the
And
beauty of this system actually.
yeah, and sustainable, obviously sustainable, because you are not generating any new kind of variation, but you are mimicking a variation that is already present in nature. Suppose what I gave you example in wild rice allele, that is present in nature, but you cannot bring it to your cultivated one for your benefit. But this technology help you to bring that allele to your cultivated one for your benefit. Yeah.
So
There is,
one
right,
question
there
from
is,
YouTube is there. So, is there any changes in the genotype or phenotype of the host by editing a defective gene?
Okay, genotype, as if I understand correctly. So suppose if you change a nucleotide or a target region, that gives you a detectable phenotype. Like I told you, WSL5 and Z3, these two genes, if you change that nucleotide, then you get the phenotype. Now, for example, as I gave you example of SWIT gene, to edit the promoter, okay, to inhibit the binding of the tail effector from the bacterial blight pathogen. So that when you edit that kind of promoter sequence, that does not give you any phenotypic difference. Only trait you get a resistance power to bacterial blight pathogen, but not any phenotypic differences.
I
So,
think,
the same question
yeah, same
Vishwajit Sahu
question
is
actually.
asking.
Right.
So, can we say genome editing as a mutation?
Yes, yes, 100%. So it's a very easy way to mutate in a targeted fashion. Suppose previously, before genome editing came into picture, we used to mutate our plant by chemical mutagens or gamma rays, I mean physical mutagens. So we used to treat our seed with mutagens and mutate all the seeds, and it is randomly mutating the whole of the genome. And then we go for selecting selecting a plant based on the phenotype that we are looking for. Then we go for the genotyping. Oh, what kind of change happened? Okay, so that was a random kind of mutation. You cannot control where it is going to mutate, the gamma ray or the chemical mutagens. But this CRISPR-Cas9 is targeted to your gene that you want to mutate. It's not a random mutation like the physical mutations physical mutagens and chemical mutagens. So it is very targeted mutagens. Yes.
There is one more question in this Zoom panel. This is Dr. Amrita has asked, how base exciting or
Editing.
exciting, maybe it's editing or exciting,
Editing.
I don't know what,
It's
is
editing.
different from site-directed mutagenesis?
Okay, that's a very interesting question. Site-directed mutagenesis, you could perform very easily in vitro, like in a plasmid, then you enter that plasmid to bacteria. That's easy. But site-directed mutagenesis in plant or in eukaryotic organism in vivo, it was almost impossible. In plant, site-directed mutagenesis have never thought of. But now CRISPR-Cas9 can do site-directed mutagenesis. Site-directed means that you want to mutate. That is possible now in vivo genome editing. In plant, you can perform that site-directed mutagenesis, not in tube. Okay. Yeah.
I think there are no more questions, Dr. Molla. So I would
Mhm.
like to thank you from our whole organizing team. Thank you so much for giving this wonderful talk and very informative talk.
Thank
And
you.
I
I
hope
have
all.
one question.
Yes,
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 yes, yes.
please, sir,
Yeah,
please.
please.
Hello, it is very nice talk.
Thank you.
Congratulations.
Thank you.
Basically, I want to know what are present guidelines,
Mhm.
whether it is considered as a transgenic only in India, or it will be if variety get released through this genome editing method, or we can say base editing method. So how it will be treated in India presently?
Okay, so yeah, that's a very relevant question. So if I want to start from USA, and then coming to India. So USDA is not regulating any of the gene edited crops or organism that does not have any foreign DNA inserted. So I think you have heard about the non-browning mushroom. That is also developed from the lab where I worked, Yinong Yang's lab. So they have got a green signal to go to market because one or two nucleotide change due to genome editing, that's kind of natural variation. It always present in nature. As you told that from rice SNP-seq database, you told how many crore SNP?
Exactly.
So this is a huge amount of differences in single nucleotide. Okay, so when you artificially create some single nucleotide differences for your benefit for agricultural purpose, they are not going to regulate it if it is not present, if it does not present any foreign DNA. So remember, if it goes through HDR technology, the homology directed repair that I mentioned, then you have to supply a donor DNA like that. So if you go through that pathway, then it is regulated as transgenic. But if you go through base editing, if you go through the normal knockout CRISPR-Cas9, it is not regulated. So there are at least 20 crops that has been deregulated by USDA. And now India, DBT is the I think MOEF, GESC, and DBT. They have drafted a guidelines, and it has also almost mimicked that kind of regulation. Suppose you are working with PTG system, your if you could develop a blast resistant rice variety, it would not be regulated as transgenic.
That is really nice, actually.
It would not be regulated. But suppose if you use a HDR technology and you transform donor template and that is integrated, then it is
It will
regulated as transgenic.
obviously.
So,
Yeah, yeah.
but it is not final. It is a draft regulation.
Mhm.
Yeah.
Thank you.
Yeah, thank you, sir.
Dr. Solanke for asking the question. Okay, I think Dr. Vinod, we don't have any more questions, so I would like to
Okay.
thank the speaker and all the participants and from the organizing committee. And I think the last concluding remark will be from Dr. Vinod. Back to
Yeah.
Vinod.
Yeah, so I would like to thank Dr. Solanke and Dr. Molla for their nice presentation and accepting our invitation. And the presentation are very nice and very informative. And people have more questions. And if anybody has more questions, so you can also email to us. We will be addressing. So by contacting the speakers also, and speakers will always be ready for answering them. and we are very much thankful to the speakers for their nice talk. And thank you all. And we are going to close this today's second session. We will be again coming live. So tomorrow 9:30 a.m. sharp. So please join us. Okay, thank you. So we are
Okay.
closing it. Okay,
Yeah,
for
bye.
today. Bye bye, and
Good
have
night.
a good night.